Good afternoon, everybody, and welcome to today's ParaBoss webinar. Today, we are going to be talking all about liver fluke. And for those of you who are new to ParaBoss, I'll take us through a little bit of housekeeping. But to get us started, my name's Fiona MacArthur, and I am a member of the ParaBoss Extension team. And shortly, we will be joined by Nichola Calvani, and she's from the University of Sydney, but I will introduce you to Nichola shortly. ParaBoss, if you're new to ParaBoss, is a joint initiative of AWI, Meat and Livestock Australia, the University of New England and Animal Health Australia. And it's really your go-to for everything to do with parasites in sheep and goats and cattle. Good afternoon.
Excellent. Thank you so much for the brilliant lead-in, but also the invitation to present today. Thank you. I'm a parasitologist, which I think means I'm obliged to teach you a bit of a life cycle about parasites, and in this case, liver fluke. And unlike our gastrointestinal nematodes, they are a little bit more complex, and that has impacts not just on our ability, on the seasonality of the parasite, but also on our ability to diagnose and manage the parasite. So just if we want to choose a starting point here, we can imagine the first stage of infection is when animals become infected by eating what we consider cysts that are found on grass, particularly around waterways, because these parasites have an intermediate obligate snail host that is an aquatic snail. So without that intermediate host, you cannot get the completion of the life cycle of this parasite, which does mean that we can't transmit it to each other. Animals can't transmit it to each other. They do require these intermediate snail hosts, which shed what becomes an infectious cyst in and around their aquatic environments. Those cysts then emerge. The parasites emerge in the guts of the intestines of their hosts shortly after they've been ingested. And the young immature flukes will spend not much time actually penetrating the intestinal wall of their host. And then they spend about 2 to 3 months actually just migrating around the liver like little tiny Pac-Men, chewing their way through the liver parenchyma before they mature into sexually mature adults that are able to reproduce and lay eggs, which occurs in the bile ducts and the gallbladder. So really what is happening is the majority of that pathology is occurring very early on in infection, but that is also occurring during the time where it's difficult to diagnose infection with this parasite, because it takes, as I said, 2 to 3 months for them to start laying eggs, which are then shed into the environment in feces, which is our primary form of diagnosing this parasite. So as you can see, it has an intermediate snail host, which many gastrointestinal nematodes don't have. It has a very, very long life cycle. So that's about 3 months just inside the mammalian host, not including all other aspects, but very associated with water. And that makes it quite difficult to diagnose. So if we continue on with our life cycle, what happens after the eggs are shed into the environment is if the temperature and climatic conditions are right, these eggs will develop and hatch in about a week. And the infectious stage that comes out of the egg very rapidly has to infect its snail host. And about a month or 2 after that, again, if the climatic conditions are ideal, these hosts, the infected snails, will shed those cysts back out into the environment once again. So as you can see, quite long and can present some really important challenge for us researching and working on ways to control this parasite, but also as producers who are looking to manage this issue in their herds. One thing I'd like to point out just quickly is, as I've shown here in this image, this is a parasite that can infect most mammals. So it obviously infects our livestock species, but it can infect wildlife such as kangaroos. So very, very hard to manage on your properties if there are animals coming through over fences that are infected. But also it's important to note that it is a zoonotic parasite, so people can become infected. And this is what the infectious stage looks like. These are the cysts that adhere to vegetation around the water level. You can see them with the naked eye, but they are quite small. However, I wouldn't advise that you look for them because this is the stage that is infectious to you. So not a great parasite to manage. As you can see already from this, it's complicated. One of the first questions I was posed is, where are liver fluke located? I think that's a really great question because I would also like to know the answer. I guess what we might call the grandfather of liver fluke research was, fortunately for us, based here in Australia for much of his career, and he produced an extensive body of work that does include distributions of this parasite in across the country. And so I've included some of his original work here, which is available from the Prime Fact 446. That's Dr. Joe Bore's work and legacy. So you can find this Prime Fact by scanning this QR code if you'd like, and I'd recommend you do because there's quite a lot of useful information on it. Unfortunately, though, we haven't had the ability to update much of his work since the time that he first did it. So this is what we expect the distribution is. This is the most current, widely distributed data for this parasite, but obviously a little bit out of date now, and particularly with some variations we're seeing in the climate at the moment as well, and those massive, flooding and rain and fire events that has potential to impact the distribution of this parasite. It's the time that we kind of had an update, I think. Some of my colleagues in Victoria have done some of that work for us. So here is some of their latest work where they were looking actually not just at the distribution in certain irrigation areas in Victoria where we do see lots of problems with this parasite, but also they were looking at drug resistance. And I'd invite you to actually look their paper up further to get some more detail about what this graph is showing. But effectively, they examined 83 herds across the 6 main irrigation regions of Victoria, and they found that there was about a 40% prevalence in those animals. And of the herds that were tested, 46 and infected, 46% of those were likely to be infected above a threshold that was impacting their production outcomes. I also, in preparation for this talk, had a look online at what's been going on more recently. As was mentioned, I've only returned to Australia at the end of last year, so I'm still getting my fingers back on the pulse in terms of what's going on in fluke land locally. But I was pleased to see that there's an MLA project that started in 2021 to look at the current distribution and management options for liver fluke in cattle. And if you go to this QR code, you can find, again, this information. It's been coordinated by Miss Leah Terrell, who is currently at the University of Melbourne working on the McKinnon project. And so far from their summary, they're seeing sort of similar trends where between 40 and 60% of herds are infected. And those percentages can range within a herd from 10% of animals up to 100% of animals infected in a single herd. So obviously still quite a significant problem in Australia, and I'd be really keen to see the outcome of this project when it's available. In the meantime, though, I've come back and I've also been asking the same question. So we've kicked off a project this year that is being conducted by Simi here. She's a third-year vet student. And as part of their third year, they're required to do a research and inquiry project. And hers, we cooked up together trying to answer some of these questions. And it's called Fires, Floods, and Fasciola Hepatica. And this project is work being done in collaboration with the Elizabeth MacArthur Agricultural Institute here in New South Wales. And effectively what we have done is collected 5 years worth of diagnostic data that has come through EMAI. And Simi is going to be providing a map of the distribution of this data through New South Wales from the last 5 years. And she's also going to look into the impacts of some of these extreme climatic events. So obviously the fires that we had, the Black Summer fires, and the recent rains and flooding on the between year seasonality of infection. So what happens between 1 year and the subsequent year, and how major these events may be impacting the distribution of this parasite and the seasonality of it. But she'll also be aiming to hopefully supplement our existing information on within year seasonality of infection. Because it does have quite a long life cycle, it is very seasonal, depending on where you're located and the climatic conditions associated with where you're producing your animals. So we're going to be very keen to see the outcome of that project. She's just finalised collecting all the data last week and is in consultation with our statisticians to begin analysing it shortly. So maybe if I come back in a year's time, I'll be able to give you an update on that. And hopefully we'll be able to produce some really current, really good data on what we're seeing, whether we're seeing problems with this parasite in New South Wales. Now, I'm just going to return to the life cycle for a moment again, because I wanted to actually provide an example of what I mean by this parasite being seasonal. So if we start with step 4 here, which is the eggs hatching, what I have done is gone and I've taken a climatic graph for Goulburn in New South Wales, which is an area where we have had reports of significant problem with fluke in a sort of sheep or a merino stud area. So obviously big impacts on sheep there. And if we have a look at this, we have some biological parameters that we can include that we know about this parasite to help us predict what's going on. So this is sort of a back of the napkin estimate, you could say, of liver fluke seasonality for Goulburn. So we know that eggs begin to develop and therefore get to a point where they can hatch when the weather is above 10 degrees. So we could say that for this region, we're sort of seeing that occur around October. However, if there are snails that are on site from the previous year and the snails are very good at hiding away when the climatic conditions are not favorable for them, they can very quickly recolonize an area after a period of dry weather or if it's too cold. So we assume they're already on the property and available to be infected, but that they're not becoming infected really until the climate is right for those eggs to start developing and hatch into the environment where they are found. The snails can then come into contact with them. So a couple of months, 1 to 2 months after that, those snails start to produce these metasaccharia or saccharia, which would go on to become metasaccharia, the infectious cyst stage of this parasite that's infectious to us and our stock. So that would be occurring a month or 2 later from that sort of 10-degree threshold. So our stocks then become infected. They take a few months, so 2 to 3 months, to start producing eggs, which then are available on pasture to infect those snails, and the cycle completes itself again. But depending on the suitability of the climate for the snails, we do get a pause in that cycle. So for these poor farmers, what that essentially means is they can effectively consider that their stock would be getting infected on New Year's Eve. We are working with 10 producers in this region at the moment on another project, which I'll introduce down the line, to actually see if this back of the napkin estimate is correct. But so far, all of our diagnostics seem to be aligning with that expectation. And I'd just like to briefly introduce you to the Frederick here. Frederick is one of the parasites we've produced as a result of our in vitro work, which again, I'll introduce at the end. So as I mentioned, the pre-patent period, which is the point from them becoming infected to the point where we can find them in the gallbladders and bile ducts of the hosts and when they're shedding eggs into the environment in their feces, is 2 to 3 months. And in that 2 to 3 months, they're very, very busy trying to become big and strong. So they start off really small. They're about 1/10 of a millimeter long. And this is one example of that. So this is a scanning electron microscopy image of a brand new baby hatched out liver fluke. And then 2 to 3 months later, this is what they look like. So I hope you're not squeamish. I probably should have warned you slightly earlier. But these are fluke being squeezed out of the bile ducts of a sheep that was infected for vaccine purposes. And as you can see, they're big and juicy and strong parasites that have spent, as I said, 2 to 3 months just carving through that liver tissue, which the poor sheep had spent a lot of time obviously trying to repair. And that's where we're getting the majority of our production losses actually occurring, is in that time. Sorry, what are we doing? Yeah, all right. So overall, there are we consider there to be 3 stages of disease with liver fluke. There's either the acute stage, where you can get really sort of significant losses very early on. We have subacute and then chronic. So we see chronic infections more commonly in cattle. So cattle can mount a bit of an immune response against these parasites. So they tend to have those losses that are very low grade and persistent over time, and therefore really important in production animals like dairy cattle, for example. This is a very known problem in the dairy industry. And you can see this low grade persistence and inflammation and irritation has resulted in those bile ducts becoming really big and thick and huge. And there's a sort of fluke there in that image in the bottom left there next to the ruler. So this is what we call this clay pipe bile ducts, and it's very easy to spot on post-mortem examination of the liver. Here we're sort of seeing a more subacute type of infection that we would see in sheep. And this is where we get these tracks and tags occurring as the little fluke are migrating, immature fluke are migrating through the liver tissue, and the liver is trying to heal that damage. You can get very large losses at the very acute stage of infection in sheep in particular, if you have if the animals have come across a large source of infection. So often, counterintuitively, that's seen during drought. As our water sources shrink, our snails become more concentrated in a region, a smaller area, and they start to produce the same number of snails are producing the same number of infectious cysts, but that area is now concentrated, and the animals as well are searching for water. So they are seeking out these areas of high infectious burden. And so you can see very sudden deaths in sheep in particular, before the pre-patent period has finished, due to the impact that is happening as the large numbers of infectious parasites are migrating across the intestinal wall and through the liver and doing the damage you see here. Generally, it is considered that once you reach 25% prevalence within your herds, you're starting to see production losses. They are very, like many parasitic diseases, they're very difficult to tease out and specifically quantify because they are quite broad, and many other factors contribute to this. Obviously, you're seeing things like liver condemnation and sudden death in sheep in particular. You can get secondary bacterial diseases such as black disease, but that's not very common these days. We manage that with our Clostridial drenches. But mainly what we're seeing is chronic losses, so associated with reduced growth rates or fertility, reduced production outcomes, but also the cost of control is a significant one as well. So I actually had a look again at some recent data about some recent information published about what you can find out at slaughter. And there was a large study done not too long ago in Uruguay in 2016, where they actually had a look at about 30,000 bovine livers from nearly 1,000 farms within a given year. And similar to what we see in Australia, despite being on the other side of the world, we're getting about a third of animals infected, and at least nearly 80% of animals of farms tested had at least one positive animal on postmortem inspection. In this region, 81% of liver condemnations at the abattoir are due to liver fluke, and that's estimating to cost producers $2.60 US per liver. But I think the more important messages we can draw out of this is that liver flukes are very good at surviving in a host, particularly if you don't know the infection is there and it's a long, low burden, long-term chronic infection, which means it can be very easily introduced onto your property if you have the right snails on your property, which means that we always, of course, are going to see more older animals infected than younger animals because they're exposed to more of the course of their life. But what this study found is that animals are more significantly affected by this infection at a young age, particularly animals in the 23 to 30 month age group. On average, they were the most significantly affected compared to the uninfected animals. And on average, that the burden resulted in a 6 kilo weight difference between infected and uninfected young stock. One other aspect that can be considered as well is that because they're putting on weight more slowly, they're potentially taking longer to reach slaughter weight, but also there are associated impacts with time to first estrus and production impacts, such as what we can see from the MLA report that was released in 2022, a 2% discount on wool value because of the impacts on staple strength. So that's sort of one aspect of data we can see at slaughter. When we look at an Australian perspective, as I mentioned, the MLA priority list for endemic diseases for red meat industry produced an update in 2022, which I always love to see, especially for liver fluke, because although it ranks sort of lower down on this top priority list, and those production impacts seem smaller relative to other groups, it is also one of the few parasites, or the few anything, that is caused by a sole species. So with the internal parasites production impacts, that's obviously up near 700 million in sheep at the moment, but that's all internal parasites. liver fluke alone is estimated to cost the Australian sheep industry, not even considering the beef industry or dairy industry, but just estimated to cost that industry 38.2 million, which has increased from the last time that they did this report. So it's a fairly significant parasite in and of its own right in Australia. But we can also get some other really interesting data at slaughter. And this is something that I was quite interested in recently. It came out not long ago, last year or even I think this year. And this is a study conducted by some of my colleagues in Brazil over about 20 years. And what they did was they looked at changes in land use, particularly associated with the Amazon, obviously with all the deforestation that's occurring there. They then looked at slaughterhouse data and correlated changes in land use with an associated change in cattle distributions. So the graphs go from A, B, C, D, so top two, bottom two for each different set of graphs. And so it's 2002, 2008, 2014, 2020. And as you can see, as we're increasing the deforestation in Brazil, we're seeing an associated change in cattle distributions moving up through the north of Brazil. And then that has caused an associated change in liver fluke distribution. So previously at 2002, when this data started being collected, Fasciola was pretty much nowhere to be found in anywhere outside of the south of Brazil. And now you can see that it has not only moved up through central Brazil, but also up into the northern regions where the Amazon is located. And so there are potentially very concerning impacts, not just on the infection of cattle and stock in these regions and the movement of this parasite, but because it's zoonotic, there's potentially impacts on people in these areas, especially native tribes. And on top of that, there are unknown consequences for wildlife in these parts of the world as well. So there's a lot of information you can get out of slaughter data, but it's a very bleak picture. And unfortunately, it is really difficult to tease out individual values for the impacts on the bottom line. So again, a great question that I was posed: resistance. What do we know and what is still a mystery? I also would like to know some of these answers. So resistance to triclobendazole in Fasciola which triclobendazole is our frontline drug, it's our first drug of choice, was first detected in the world here in Australia in 1995. And in that time since, we haven't learned a lot necessarily about how this drug works and therefore how to overcome resistance, or how resistance works and therefore how to maybe get different drug targets, etc. So I'm just going to present you with a little summary, and I will point out that this is probably the area that I have the least strength in. Pharmacology is not a strength of mine, but I do have lots of colleagues who are fabulous and able to help with these sorts of things. But effectively, these are the drugs that we have available worldwide against liver fluke. Not all of these are registered in Australia. And these are the stages at which they become active post-infection in our stock. So as you can see, triclobendazole is that one really rare one that is able to kill stock in that very, very early infection period, which is when they are starting to cause all that damage. Unfortunately, resistance to many of these drugs has been reported around the world. And we still don't really understand the genetics that underpin this resistance or how this drug actually works on the parasite. And that scares me in particular as a researcher that works on this parasite, not just in stock, but in people, because triclobendazole is the only drug that is currently registered for use against this parasite in people. And there are many parts of the world, particularly in Southeast Asia, where I did my PhD, where this parasite is considered hyperendemic in human populations. And there are many school children, for example, that are impacted with this disease. So I am particularly concerned, as I'm sure many people in the Fluke research field are, about this drug resistance, but unfortunately, we don't have lots of answers available just yet. There are dedicated teams working around the world on this problem. So this team in Liverpool is renowned. They were the first people to publish the genome of Fasciola. They're renowned for their work in trying to understand the genetic basis of resistance and understand and identify targets that this drug works on. And they published this paper last year. So they are narrowing down in on the answer, but we don't have anything definitive just yet. So I too am interested, as I said, in this in Australia locally as well as internationally. And so we have a project as well that's happening this year called Beyond Suspicion. After being contacted by some producers from the Southern Tablelands of New South Wales who have a livestock, a stocking agent who's very interested in, he does fantastic fecal diagnostics and monitoring, and very interested in sustainable control of parasites over the long term. He's been doing it for about 20 years now. And he was concerned that he was seeing an increase in fluke egg counts of about 250% over a period of a few years. So what we have done is teamed up with him and 10 producers in the region who have concerns over fluke in their animals, and we will be testing them. So this is where we'll be able to confirm or deny some of that back of the napkin estimates in terms of the life cycle that I mentioned earlier, because this project is based in Gunning near Goulburn. And we will also be testing the efficacy of not one, but 2 drugs in these properties to be able to provide an update, because we don't necessarily have that information for New South Wales, and it's about time that we get that. So that's what Chelsea will be doing, and she's supported by a very robust team of veterinarians, livestock scientists, epidemiologists, and we have a representative from Elanco Animal Health there as well, helping us with the pharmacology aspect of the project. So that's, again, something that we'll be able to update it in 12 months' time. Now, I always get asked, so what do we do? And when I present to large audiences such as this that are from all parts of Australia and even internationally, I would generally err on the side of caution. I can't give you an answer unless I know specifically what's happening on your property. But in fact, WormBoss does have a very useful website to provide you with the most recognized and available information we have currently. So if you go into WormBoss, you can look up liver fluke and you can get results for liver fluke in cattle and sheep and goats. And they have strategic treatment suggestions where they outline what is currently recommended. So I would very much suggest that you check that out. But when you're doing that, please make sure that you're thinking holistically about how you're going to control this parasite. Drugs are not the only answer. And when you are using drugs, it's really important to use the right ones at the right time. So make sure you're getting drench tests done to know that your stock are infected and that your drugs are working. And also try and do your own back of the napkin maths, I guess, based on these slides on when your area should be seeing issues with fluke, and then try and time your treatment strategically around that, keeping in mind that we really do need to preserve the activity of triclobendazole as much as possible. So that is essentially my advice for the using drugs to stop treatment, but there's not a single pronged approach that we take. As I said, the snails are really the problem. I've got 2 snails that I'm going to introduce you to here. So the one on the left there is called a planorbid snail, and that is the intermediate host of the rumen fluke parasite or stomach fluke parasite that you may have heard of, which there is some debate at the moment about whether or not they're causing pathology and disease in stock. But they're the hosts, and they do occur in very similar regions to the limnaeid snails, which are the intermediate hosts for liver fluke. But the 2 parasites, the 2 snails do look very, very different. Now, this figure, I use this image a lot, although it's a horse, it still shows some good tips, or very succinctly shows good tips for managing those interactions between snails and the infected stock, and that is by fencing them off. Now, we are not talking about liver fluke in horses, so we'll introduce a cow to this scenario. However, this is how I used to present these slides before I went off and lived in Ireland for 3 years, where especially on the west coast of Ireland, it rains every day. So this is the environment I was spending my time in for 3 years. And if we have a look on the other side of this fence, this is a perfect fluke environment. And there's not a lot that you can do in an environment such as Ireland, where there is so much water on the ground all the time, all year round, to prevent this from fencing by fencing. So I know that is easier said than done, but I must stress that it is your biggest key to trying to control this problem in your flocks. Just a quick note on the snails, it's not really worth trying to kill them and stop them. They reproduce very, very quickly. It doesn't take many of them to colonize a region, and they do it very fast. They can actively migrate themselves, either by moving, or they can passively migrate by being trodden through on stock hooves, etc. And as I said, they can survive between seasons. So they're really hard to control. And there actually was an MLA report that advised against this, published in 2003. So what I'm going to tell you to do is, if you are putting in the efforts to try and control this property on your farm, make sure you are putting in your efforts in the best way possible. So having have access to dams, particularly if those dams are then fed as irrigation into other paddocks, is a really bad idea, because that's giving your stock and the snail hosts a perfect opportunity to come into contact with one another, and that's going to perpetuate the life cycle. This too, I do advise using water troughs, but don't do it like this. I know that's, again, an easy thing to say, but as you can see here, even something as small as one little leak can cause boggy areas to build up, which are the perfect environments for the snails to replicate in. And that is where your stock are going to come into the most contact with them when they're drinking and defecating. So that's also not great. It's important that we minimize these interactions, because it doesn't take much for one snail to become infected with one single Miracidia and then produce 4,000 of those infectious cysts as a result. So it's really, really key that we try and minimize those interactions while maximizing the efficacy of the drugs we have available to us. Just a really quick note, I know we're wrapping up now, but just a quick note on my other research activities in this space. So I have recently, as I was mentioned, been awarded a DECRA for 3 years to study specifically host-parasite interactions in the lab. So this is very exciting, because it's the first time we've really been able to actually watch what happens when the parasites come into contact with host tissue. And we were very surprised to see some of our initial results, that they are actively feeding within less than 24 hours of emerging in the guts of the infected hosts. As we can see here on these videos, this is liver fluke, less than 24 hours old, feeding on fluorescently tagged tiny little livers in the lab. And you can see that they're not only migrating through those little livers very quickly, but also, as I said, ingesting and digesting these cells within 24 hours. So that's a very exciting frontier. And all I can say about that is to keep watching. There's lots to come from this space as well. So I think I'll wrap up there, and was just wondering if there are any questions.
Thank
you for listening.
That was a great overview there of liver fluke, and it was really good to get an insight to what's going on with research for industry as well. So thank you. That was a bit of a treat. Now, I did get a little bit of a glimpse at your PowerPoint beforehand, so I do know that you have some additional slides, and I have one early question that's come through, which I think your extra slides might be helpful for this one. But before I give that to you, this is your opportunity now, everyone, to put your questions into the Q&A panel, because once we finish with this question, if there's no more questions, we'll wrap up. So don't lose the opportunity to ask whatever questions you like. So the first question I have is, when testing for fluke antibody levels using a blood ELISA test, how long will this test show a positive result after an infestation has been removed? Yeah,
that's a really great question, and I'm glad I added those additional extra slides in. So let me just quickly bring those up. And I'll just close this off so that's not in my way. Okay, so yes, there are 3 options for diagnosing liver fluke at the moment as it stands. The first one is the cheapest and the tried and tested true gold standard for the industry, which is the sedimentation and a fecal egg count. That is looking for those eggs that the parasites are shedding in feces. And it is great, but it is limited in that 2 to 3 months when there is wandering around the host. You can't detect a patent infection, so an active infection, using that method. So 2 other options that are available are the serological ELISA, so looking for antibodies that are produced by the host, so by me or by you or by the cattle or sheep, in there, and those antibodies can be detected in their blood, but also in milk, which makes it a really great surveillance tool for dairies. And the third option is the coprological ELISA, which looks for antigens that the parasite is shedding into its host, and detecting infection again through the feces. So these 2 allow detection inside of that pre-patent period. The earliest point of infection that you can detect using the ELISA is, we generally say, about 4 weeks. And with the coprological ELISA, we say it's really about 6 weeks. But the question was asked, how long after we treat should we be able to can we use the serological ELISA again? And that's a valid question. And I'm not sure if I have the exact answer on my slides. Let me check. Nope. So I'll just talk through it. So effectively what happens is if you treat an infection for liver fluke with an effective drug that kills 100% of the parasites, those parasites have died. So they will not be shedding eggs in the environment anymore. They will not be shedding the antigens into the feces anymore. But it takes some time after treatment for the antibody levels, so the thing that's produced by the infected host, to decline, which is a good thing and a bad thing. So it's a good thing because we want to exploit that persistence of the antibody response when we design vaccines. But it's a bad thing because it means that this test, the serological ELISA, we effectively do not consider that a diagnostic test in anything older than 1 year. So effectively it takes, and it depends. So I worked in Ireland in the lab where we sort of the first antibodies and proteins were identified for the development of these tests. We spent a lot of time doing these ELISAs, and we recently published a paper where we tested pre and post treatment and compared. Now I have to warn that was under an experimental condition. So that means we infected the animals on a certain date at one point in time. We tested them over several weeks and months, and then we treated them and then continued to test them. But they only had one infection. And it does take sort of several weeks to, I would say, 2 months for your antibody levels to come down below what we would consider a diagnostic threshold. But that is also dependent on the fact that your animals are not becoming reinfected. So they're not having their antibody response stimulated again by new infections. So we don't consider the serological ELISA diagnostic, but it's a great tool for if you are trying to surveil your stock. So if you, for example, it's great in dairies, particularly in heifers and a young stock. So if it's their first season and this is following a very seasonal infection on your property, it's a really great tool to surveil groups of animals. You can test 10 animals at a go in one test, and it gives you some idea of when your animals are becoming infected if you do it, every couple of months. But it's not what we would consider diagnostic, because if at any point your animals become infected post-treatment under what we call a natural setting or a trickle infection, those antibody results are going to go back up again. So they take a long time to decline, and we don't consider it diagnostic. I hope that helps.
I'll follow on with another question about ELISAs. Do you have any thoughts on how to interpret ELISA screening? For example, this person recently bled 10 sheep for 2 times pooled counts, was soon to be joined composite maiden news for fluke presence. 1 pool came back with a very low positive. Do you think an estimated guess of less than 25% prevalence warrants no specific fluke treatment?
Yeah, okay. So I'm just going to try and interpret that result step by step. So effectively, you bled 10 sheep and you split those samples between 2 pooled counts. Now, do you mean the ELISA result or the fecal egg count? Because an egg count is different to an ELISA. However,
it says ELISA screening there.
Yeah. So effectively, if you've tested 2 groups of sheep and you've combined 5 samples, that's actually really giving you an idea of what's happening in two populations. So for the 25% prevalence threshold, you'd want to be screening larger individual numbers. So not pooling your data, but actually testing it individually. And remember that 25% threshold is for within herd prevalence. So if within your herd more than 25% of your animals are infected, that's when you'd be expecting to see production losses. Where'd it go? Because I wanted to answer one more thing. If you're saying you've had a low result though for the ELISA and only in one of those 2 groups of 5, if that's something that's happened sort of recently and a young stock, I'm not surprised that your ELISAs maybe are low at this time of year, depending on where you're based, because this is when we would start in southern New South Wales, for example, to see animals become infected and start to increase their results, positivity of their results. So if that's been any time in the last 3 months, it's worth getting another test to see if that is continuing upwards. But if it's only sort of a low result in that pool of animals, it potentially means that not very many of them are infected as well. So the ELISA results should be interpreted with care. They're a great screening tool to inform when your stock are becoming infected if they're young stock, but I wouldn't be making treatment decisions based on the results of the ELISA. I would always want to follow up with a fecal egg count because that gives us an idea of when our infections are becoming patent. So that's when they're shedding eggs into the environment.
Great, thank you. Great advice. And next question, is there any potential to select for host resistance?
Yeah, that's a good question. I know that resistance and resilience is something that's often talked about in terms of livestock and parasites. I don't believe there have been many. So, for example, the AASBVs, the Australian Sheep Breeding Values, I don't think there's been much work done in the scope of looking for resistant traits for liver fluke. I know that that's been done in terms of like DAG scores and whatever for the gastrointestinal nematodes. I'd have to say that this parasite is a really great parasite. So it does infect almost all mammals. And there are some species level differences between their susceptibility. Specifically, you know, cattle are more adept at mounting an immune response and getting rid of their infections to the point where they just have sort of low grade infections. Sheep are much more susceptible than cattle to higher grade infections, and they're much more vulnerable to those infections. But even sort of within sheep, we see differences in resistance profiles. And I'm not currently aware of work that's looking into determining whether or not we can breed for resistance, but that's something that I can check up on.
I'd have to do that webinar in another 12 months, I think. There's lots of things.
I gotta do some homework.
Yeah. Next question. This is your first research project that you mentioned about the drought and fires and its effect on liver fluke. How do you think the Brazilian research on landscape changes and liver fluke prevalence will relate to changes in nature of the Australian farming landscape and practices?
Yeah, I mean, that's a really great question. I can't forecast, I don't know, but we are, as I've just said, I've come back to Australia and I've got the same questions you all have, and I'm trying to work really hard to see what we can answer as quickly as possible. I would be very interested long term, so over the next 5 to 10 years, to start tracking this data as well locally and see what changes on land management are doing to this parasite, because I think this parasite acts as a very good red alert because it is zoonotic, so people can get it and it has impacts on our native animals as well. Kangaroos are very, very susceptible to it. So it would be interesting to do something similar here. But we don't have that information available yet. Now, I hope I'll be back in 12 months' time with some interesting results to talk to you about in terms of what impacts extreme climatic events are having. As I said, the fires and floods, but changes in land use is something that has yet to be investigated in the local context and is certainly something we're really interested in.
Where are we up to? Producers have mentioned on some reports that they've got back, and I'm assuming this is from the abattoirs, with lung hydatid cysts. This person was under the belief that it all was just from the liver. Do you have any comment there?
Yeah, so again, hydatids, they're a big problem because they are zoonotic, and they obviously have big consequences for condemnation of carcasses. They can occur in the lung as well as the liver.
Thank you. With the liver damage, that occurs from the juvenile migration through the liver prenapia, I hope I pronounced that right, how permanent is this damage? Can this damage cause lifelong ill thrift that is visually obvious in individuals despite eliminating the fluke infestation? Well, that's some great questions today.
Yeah, these are really good. Testing me out. Okay, so what I can tell you about the liver damage. So lucky for us and lucky for me, after spending 3 years in Ireland, the liver is the body's most organ that's most capable of regeneration. So it is very good at bouncing back from significant injury, including what is being done as the parasites migrate through. When we have done vaccine trials and when we have done drug trials before in sheep, it always surprises me at how heavily infected a liver can be, despite not showing many really obvious external signs. So if you had in sheep that you infected at one point and you killed, a couple each week and tracked that infection in the liver over time, you would see that initially, as they're first in that parenchymal stage, they're causing a lot of inflammation and damage, and the host is working really hard to repair that. But once they get to the sort of adult stage and they're in the bile ducts, that damage is quickly repaired by the host, but can cause, depending on how severe it is and the species, can cause significant scarring. What we see is primarily the production impacts are occurring in young stock up to that 30-month age point. And then it's hard to tease out, but we do think that they can recover from that, reduced body weight impact as they were growing. One thing that's important to note is just because they're able to recover from the liver damage and able to make up some weight, these parasites are really clever. What they do is they secrete essentially proteins and chemicals into their hosts that not just doesn't just keep them nice and safe and help them create a nice, happy environment to live in, but actually influences the entire host's immune system. So infections with this parasite can actually cause confusion in interpreting other diagnostic results. For example, I think tuberculosis testing is impacted by whether or not an animal is infected by this parasite, and it does skew the immune response. So even though the animals can physically recover on a gross appearance perspective, whether that be liver or weight, there are still consequences of infection even once they mature, and they're not damaging that host so directly from the bile duct.
Thank you. We're getting close to the end of our questions. We have a couple more to go. Is there a maximum temperature that Fluke will become inactive?
Yes, like everything, it's not completely indestructible. It does eventually keel over and die. It's actually not temperature that is important for its survival. The hotter it gets, the more quickly the parasite stages develop until a point, which I believe is about 35 degrees from memory, but I could be getting that wrong. What is most important, however, is the humidity. So they can survive higher temperatures if the humidity is still very high. We generally consider them capable of withstanding an Australian summer. So that is what we advise producers, is they do start to die off once the weather gets hot. They die off faster once it gets dry. But if they find a nice sort of boggy area near a trough or, in the shade, then those cysts, and particularly because they're associated with waterways and vegetation where there are many little shady spots that are usually high moisture throughout the year, they can still persist in Australian summer. That's what our general advice is.
Thank you. With increasing tribendazole resistance, what are the available effective options now?
Yeah, good question. And I am actually going to refer you to the ParaBoss website, because they do have a very great list of available formulations on the market and whether they're available in what form they're available in. So pour on, oral, injectable, as well as that. It would depend what you've previously used in your stock, and it will also depend on what other parasite problems you're simultaneously trying to manage. So I will say I'm going to handball that one to ParaBoss, just because it's a very, very complicated question to answer, and it requires a little bit more insight.
No worries. Well, there are great resources online on the ParaBoss website, and if you need any assistance, you can just email us any questions. So the last two are about controlling the snail. Is it possible to control the snails by adding copper sulfate to water sources, or use snail traps to control and reduce the number of snails?
Yeah, so there has been a lot of work done over the years looking at biological or chemical control of snails, and a lot of money, I think, and time has been invested into this, because, as you know, preventing that host snail interaction is really what's going to break that life cycle. Unfortunately, pretty much every avenue that I've seen that has been explored has been proven either not worth it in terms of a cost benefit perspective or too damaging to the environment. So I wouldn't recommend pouring any salts into your irrigated waterways or anything like that, or copper sulfate or anything, because not only is it going to have potentially bad consequences for the ecosystem in that space, but also, as I said, it takes not very many eggs for you to all of a sudden have thousands of snails again. So controlling the snail populations, the only advice I would give you is if you're using troughs, make sure that they are A, not leaking, but B, maybe sort of around those peak periods for your regions, go through and have a clean them out physically and make sure that there are no sort of buildup of snail populations in your troughs. But then I don't advise any form of biological or chemical control of the snails outside of that, because it's not an effective use of your time, money, or resources.
Great. Thank you, Nichola. Great job. Well done. That was an epic question time. And we've got some lovely feedback there as well, saying brilliant talk. Thanks very much. And thank you for the great webinar. So everyone has enjoyed it today. So it's have been our absolute treat to have you online with us. So thank you very much for joining us.
No worries. Thank you again for the invitation. And as I said, hopefully I'll be back in 12 months' time with some answers to those really great questions.
Sounds great. Thank you very much. And thank you to everybody who took the time today to join us. It was great to have your support and have you online. Remember, the follow-up survey will launch after this. And I do appreciate your feedback, because I'm looking for some final topics that you might like added to the calendar before the end of the year. So thanks, everyone. We'll see you at the next webinar. Bye for now.